Great. I'm just going to make sure that everyone can see my slides. Okay, we're good to go. So I'm really excited to be talking about this project tonight because this has been a gigantic effort by so many people at True Diagnostic and my lab at Harvard Medical School. And I've entitled this talk Precision Aging, using omics and EMR to optimize aging for each individual. And I want us to be thinking about this idea of precision aging as we are going through this presentation. And I know we have a range of people on the call, so I'm going to start from the beginning and move on to more complex things. But to just get us all on the same page, I want us to step back and ask the question, how do we think about aging? And of course, there's the idea of absolute aging, which is just an issue of time, right, and chronological age. And when we look at that issue of time, there are some people who we may look at and they may seem timeless, where time stops, right? And they seem like they look the same over 30 years, like we see here with Jennifer Aniston. And in thinking about that, while we just looked at that visually, we know that biologically, while we may all have an absolute chronological age, there is tremendous variability in what actually our biology is doing and how fast we are aging and why that is. And so that's just highlighting this idea of the difference between chronological age and biological age, and recognizing that those two are most often not the same. And in thinking of it here and in looking at these two individuals, we can think about the idea of differences in chronological age and biological age on the spectrum. Well, while we are always increasing chronologically in terms of aging, biologically, we may not even always be increasing. Sometimes we may be decreasing, but there is a variability around what our chronological age is in terms of what we are observing in terms of biological age. And of course, that's due to a lot of mechanisms biologically, and we know that there are a lot of ways that we have in our own selves the power to increase or decrease our biological age by the choices we make. Yet some of the choices we make, some of this variability may actually not be us. It may be underlying genetics. But can we understand biological age, and actually can we measure it? And a lot of what True Diagnostic and their mission is to measure it through biological aging clocks, and actually the first in class best biological aging clocks. So as we think about biological aging, and as we can recognize there, I have the two people from the figure before put here, and the woman who is very fit, who's actually in her 60s, is actually a real Instagram star. You can see certainly that while people may be increasing at the same chronological age, certainly biological age does vary. But I want to step back and also highlight this concept of biological aging and what we as researchers think of it. And I want to really highlight this point that there has been a paradigm shift in how we as researchers think of aging. And fundamentally, we think of it as a global crisis in itself. We think of it as a disease, right? That we think of aging like we do any other chronic disease outcome that progresses with time. And that in fact, in many ways, we may be able to reverse biological aging and perhaps even prevent it as well. And so when we think of it in that framework of a chronic disease outcome, and that it is slowly over time, you may increase, but there is leeway and ability to prevent it to some degree. We want to think about why that is so important. And it's simply put, because if you think of aging in that framework, is absolutely the biggest risk factor we have for any chronic disease. So this is like the master of all masters chronic diseases. And we can understand that and recognize that by just realizing that if we think of it this way, it is by far the biggest global health burden. And in fact, if we can just reduce this a little bit, we can have tremendous impact on public health. And here's some statistics. If we delay aging by 7 years, we cut disease incidence in half. And not only that, if we slow aging by just 20%, the US alone would save $3 trillion in spending over the next 50 years. So tremendous impact on public health. And so fundamentally, I myself, when I think of aging, I think of aging and biological aging as really this big, broad conceptual concept of the totality of health of an individual. And how can we understand aging in that broad framework? And what utility can it have? And while we think of it as the totality of health, I want us also to recognize that while we have a very large impact on our health, it is not just us. There's always this concept of nature versus nurture, right? And what can you do? And what are just the genes you were born with? And I will illustrate that with this example actually from my family. Here is my great-grandfather in the middle here. He was born in 1894. He lived to be 107. And by golly, if he actually did things well, like took care of himself, I don't know how long the man would have lived. But this man was diagnosed with black lung when he was under 30. He chewed tobacco until the day he died. Yet he never developed any type of cancer. He never developed heart disease. And at the end of his life, he ended up dying of pneumonia, and was running around while this happened. So certainly, this is an example of nature. There was not much nurture going on here, but nature was in his wheelhouse, and I hope I inherited those genes. So with that in mind, my point is, when we think of aging, we not only want to think about important concepts of biological aging, but we also want to think of precision aging. And what do I mean when I say precision aging? Well, for those of you who are MDs and have looked in research, right, the concept of precision medicine and personalized medicine now is really big, right? What can we do to treat our patients to optimize their health? And I want to just spin that paradigm around to aging. As researchers in aging, we can ask the question, what can we do to optimize an individual's aging? And how can we optimize it differently for one individual versus another? And in thinking about that, we can start off with a big question of, well, number one, we need to get some estimate of their biological age. And once we get that, we need to actually understand for that individual, what is driving their biological age? Because it may be different for George than it is for John. And once we identify that, can we identify the mechanisms by which that is operating? And perhaps then utilize that information to optimize and or prevent disease. And again, this is on an individual scale. So in thinking about precision aging, I also want to think about the context of which, as clinicians, precision aging is important. And of course, this is a context of thinking of this with how clinicians work every day. And what is that they use? They use the electronic medical records. But as someone who does health research and aging research, my vision of an EMR is not really what it is today, but the potential of what it can become. And while I know there are a lot of barriers to that, and we could have a lot of conversations about that, my vision of what the EMR should be is where John Doe clicks on his personal health profile and out pops an estimate of their biological age, but then personalized estimates of their disease risk, their potential response to different drugs, information about genetics, underlying genetic predispositions, other exposures they may have, things like gut health. But then most importantly, this idea of disease prevention, and that goes into all of these ways that each individual may be able to modify their disease and or aging profile to be as young as it can. And my point in showing this is that this paradigm shift of personalized medicine is exactly what precision aging is about. It's about making this a reality. And so how can we think of this now in the context of what we're talking about today? And we've talked about, there was a great opening by Varun about ideas of the aging clocks. And I want to talk about aging clocks in the context of precision aging and why what we did today is so much different than what has been done, and how we have just started what is now an explosive area of research and ways that we can add on to this to give translational potential for the patient and for the individual who wants to optimize their health. And I'm going to be talking about this in 3 different areas. The idea of creating a biological aging phenotype, and how we can predict that biological aging phenotype, and how we can understand the biological aging phenotype. Now, let's start with creating. And with each one of these areas also comes a specific framework and focus that we're going to talk about. So in the idea of creating a biological aging phenotype, I want to talk about creating a biological aging phenotype in the context of the EMR. And why is that? Well, for people who want to potentially implement something, fundamentally, if we can use data that are available in the EMR, that is incredibly important because potentially we can recapitulate this. And that is a very important framework for which to have. So for this first step, I want to talk about creating a clinically relevant and reproducible biological aging phenotype. And I put in here clinically relevant for many reasons, as we'll discuss. And in what Varun opened up with, he opened up with a brief outline of the first generation, second generation, and third generation aging clocks. And those aging clocks, the second generation and the third generation, are based on aging phenotypes. And I'll highlight a few here because these are ones that are very popular and have been, I think PhenoAge is the first one I want to highlight, which is really one of the first second generation aging phenotypes that gained a lot of attention. And what was the basis of PhenoAge? Well, a lot of PhenoAge was based on clinical data, diagnoses, and labs. And as you can see, all of those things are important to aging. And all of those things are important to aging well. And that is something we also want to talk about. It's not just about aging, it's about aging with wellness. Now, another very popular and a very powerful aging phenotype that came out or approach was simply this idea of estimating time till death. And GrimAge is a phenotype where we can estimate time till death. And that fundamentally is also important too, because we don't want to die. So I want us to think about both of these concepts, the concepts of mortality and the concept of wellness. And I want to just highlight, as Varun shouted out to, also what we aren't going to be talking about today, but certainly is an area we are going to be getting into more with EMR data, is the rate of aging. But while there have been clocks based on the time till death, and there's been clocks based on a lot of clinical labs and phenotypes, I want to posit that for the best overall characteristic of what we want, it is not one or the other. We want to live well in wellness, and we want to prolong death. So one of the questions we ask to begin with is, can we create a phenotypic clock that uniquely combines these two things? Not only can we combine these two important aspects of aging, but can we do it in the framework of the EMR, where perhaps at the end of the day, this could be implemented easily? So that's what we sought out to do, to marry these two major phenotypes into one. And what did we use to do that? Well, we used dense electronic medical record data on individuals followed for up to 30 years. And I want to point out this is important because it's also real data, right? This is the type of data we have, clinicians get and a way to track people. But can we actually use that raw data that may not be perfect in this context of creating this phenotype? And so what we used was data from the Mass General Brigham on about 60,000 people that we queried and had been followed up to this 30 years. And we decided to create this new variable where we're combining time till death and clinical and lab data in a phenotype that we are now calling EMR age. And that is creating a clinically reproducible phenotype via the EMR that balances mortality and wellness together. And now, one thing I want to highlight here, we're just so far only talking about clinical data in the EMR, but I want to keep in mind for everyone out there that when we did this strategically, and that is because these individuals that we have electronic medical record data on are also part of the Mass General Brigham Biobank. And this comes into play in the next step, but keep in mind that this wasn't just people that we chose randomly, it was people who had biosamples in the Mass General Brigham Biobank. So for this first creation, what did we do? Well, we queried all these clinical labs that were available in the EMR. And when we did this, what we focused on were the primary labs that clinicians use all the time. So we looked through the labs and it was about 50 to 60 clinical labs and phenotypes that we extracted from about 60,000 people. We had, not only demographics, and they aren't all included here, but a lot of the things from a lot of the panels, CBC panels, hepatic panels, lipid panels, basic metabolic panels. And we haven't even included them all here. And actually, as we've gone along now, we've added more and more and more panels. So we've extracted a tremendous amount of data from these panels. And at the end of the day, one thing we want to highlight is when we're doing this and we're creating this EMR age phenotype, for individuals to actually be included in our mathematical model, they have to have complete clinical data for all of the labs we ran. So just think of this for people who and you're in EMR, take a person at a point in time. Do they have all of this information at one point in time or in a very narrow point in time? You see, it's very easy to lose people. So while we had all these clinical labs over a long period of time on about 60,000 people, when we look at them at specific time points, we lose people because of incomplete data. So we actually did this whole thing of balancing out completeness with size, and we ended up with about 30,000 people. And we looked in these 30,000 people, and one of the advantages of using the EMR is you may have them at one point in time, you have all this clinical data, but there's prospective follow-up. So you can actually see who's died and who hasn't. And in these EMRs, we have very well curated data on death that we've now combined with our EMR. So we actually do know that they died and why they died, et cetera. So we have pretty complete data on that. And from this, we use the clinical labs in combination with when they died after the time that we have this information to estimate their time till death. And from that, we take all of these labs and we see which labs are important, which labs are driving the prediction of our model. And that was a selection of these 60 clinical labs, and we see them here. And that basic model where we are predicting time until death using clinical labs is our EMR age. And here you see in this model, we can actually look and see what different variables were weighted higher and what variables were weighted lower in our predictive model and our estimate of EMR age. So this created our first estimate of EMR age. And what we asked then, and what is very important to think about is, we don't just want to create any value for this EMR age. We want to make sure that what we are creating for EMR age is the most robust, reproducible phenotype that combines death and wellness together that is out there. So what we then did is we looked at how we might be able to create this in all sorts of different frameworks to see how robust it was, how reproducible it was. And here are just a few examples. We looked at our value for EMR age and we said, okay, well, for some people, if we look at all the clinical data that were included, we maybe at first we required a very stringent 6-month time window of all those different labs. But if we relax that and we said, let's just say we needed those labs within a 5-year window, how correlated would the estimates of EMR age be? And what we saw here is when we tried to reproduce these, we saw striking correlations. And this is really important because let's say you're in an EMR and you have all of the data really close, but you only have one of those values that's like far away. You want that individual, you still want to be able to estimate it. And this is actually validating that, in those cases, we can still estimate this with really strong correlation. But not only that, we then did something else. We said, okay, we want to ask this question that if I took 4 different time windows in electronic medical record data, and I took all 60 of those labs at those 4 different windows, and I created 4 versions of our EMR age model, and they're all different. One's created at 2008, 2010, 12, and 14. But then I put my information in all 4 time points. You would hope that my EMR age comes up to be about the same, right? Because if I'm putting my data in, it should all be the same regardless of what clinical data we use to develop the model. And when we did that, what we saw was really striking. We saw that the reproducibility of this model was unbelievably good, that it didn't matter what time point we did. When we created this variable, it was remarkably robust with correlations from 0.98 to 0.995. I mean, these are almost perfect correlations, really suggesting that our EMR age variable is first in class. And this is very important to us because everything we are doing from now on is based on the premise that this variable and this biological aging phenotype is the best for not being one that's looking at age acceleration. Well, we'll work on that later. So with that now, let's talk and see what we can see with our EMR age. And here's an example, and I'll explain what this graph is. I'm looking at EMR age and comparing it to chronologic age. Now, this is what's called a survival probability. So if we take each individual and we follow them for 12 years, we've divided people into groups. So if we look at EMR age and we see the orange group, these are people with our EMR age variable, we are saying are the oldest people. They are over 80 in our model. Now, we would expect people over 80 have the strongest, highest probability of dying. Whereas people who are in our youngest age group, less than 50, we don't expect them to die. And what we see is when we look at the probability of dying, or in this case, it's probability of surviving, among those who are the oldest, we say they die at a much, much faster rate than what would happen chronologically. So, for example, chronologically, by age 4, by 4 years from the time of the sample, you only see here that it's about like 22, 23% die. In our model, by age 4 years after, we're at, 35% die. And not only that, when we go out to 8 years, in our model, those who are oldest, we have 65% of people die. Whereas in the other model, we're well below, we're just hovering at 50%. So we're really showing striking that we are really able to identify those people who are dying faster. And not only that, we have perfect discrimination in those who are the youngest. No one in our model dies in the youngest group. And in chronological age, some people die, not many, but some. Now, that's great when you compare it to chronological age, but we also want to look and ask about other aging phenotypes. So when we look at this and we compare this also to PhenoAge, which is the other clinical phenotype that is out there, we also see we perform far better. And actually, PhenoAge sort of performs like chronological age. So suggesting that we really are able to identify and discriminate people in an important way. So I want to ask other questions about EMR age. We also want EMR age, it's important to us that EMR age is relevant clinically, right? We want it to be associated with different disease outcomes. And here, this is a graph where we look at the risk of these different types of disease incidences within 1 year. And we see that in all of these cases, we have, associations with all of these chronic disease outcome areas. But not only that, when we look at EMR age and we put this in a broader framework of not just one year, but a broader time span, we see here that EMR age has much stronger associations with all of these chronic disease outcomes than PhenoAge does, suggesting that it is clinically relevant. And here I've highlighted in the back that the two that seem to be the strongest are cardiovascular and type 2 diabetes outcomes on top of mortality. So our EMR age is really addressing a lot of points well and much better than some of the other biological aging phenotypes. So at the end of the day, when we're talking about just the EMR, what are we able to do? Well, we're able to work within the current clinical system and capitalize on longitudinal EMR data. And, we consider barriers to broader implementation by really focusing on things in ways that this could be implemented easily. But we are able to create a robust and reproducible EMR aging phenotype. And this combines two things, wellness and mortality together, which really are the two things we are most interested in for healthy aging. Okay, so that's talking about this area of create. I want to now move over to the area of predict. And of course, this is what aging clocks are all about. They are all about, can we estimate and predict biological aging well? And I know a lot of us are familiar with this area, but I want to talk about this area in the framework of our new OMICm Age phenotype that we are using in this predictive model, and talk a little bit about how it's been developed and why this is so important. But why it's so important is fundamentally also related to the next area, which is understanding. But let's talk a little bit about predict. Now, everything I'm talking about in the framework of prediction now, we are talking about predicting our EMR age that we just developed. And I want to step back and just ask this question, because this is, of course, a big premise of why we create prediction clocks with the omic data that we do. And that is asking this question of biologically, what's happening when we age? And we know that the epigenome is really the biological framework of aging. These molecular changes are what is responsible for our biological aging. But I want to highlight just very briefly what that is in an analogy here to make sure that we're all on the same page and understanding what exactly is happening. And I want to give this analogy with banana bread, where over on the left here, we have the ingredients of banana bread. And on the right, we have the recipe. So imagine that the ingredients are genetics and the recipe is epigenetics. Now, the ingredients never change. The ingredients are the ingredients. The ingredients are your genetics. And they are my genetics, and those are staying the same. But with aging, what is happening to the epigenome? And over time, what is happening is you can think of it as if you took this banana bread recipe and slowly over time, parts of it start getting difficult to read. And as things are getting difficult to read, the ability to make the banana bread becomes less and less. I mean, you're still making banana bread, but what I'm seeing in the recipe right now and how to make it isn't going to turn out quite as well as when we had the full recipe. And this idea of alterations to the epigenome causing our biological aging is really fundamentally what is happening. And ultimately, this may cause unreadable parts of the epigenome, and that causes aging. So I want to highlight this because if we now think of this in the framework of epigenetics, we can think of this, and we may take the epigenome of a 20-year-old and a 60-year-old, and you may see that what's happened from 20 to 60, particularly for DNA methylation, which is a type of epigenetics, is that there are these changes in methylation, where in some cases things may go from hypermethylated to hypomethylated. The main point is these may be changing up and down, but they are changing. They've not stayed the same. And these are examples of where the recipe isn't being read right. And so now the level of methylation is off. Now, what has happened in the field of aging happened, well, quite a while ago, we recognized this association between epigenetics and aging way back to the 1960s. It was in, about 10 to 12 years ago that Steve Horvath was a senior author on one of the first papers demonstrating the striking association between DNA methylation and chronological age. And I remember sitting in the American Society of Human Genetics when he first presented this, being astounded by this correlation. And of course, now it is a whole field from this has exploded. And here we see in that initial paper, this strong correlation with chronologic age and your DNA methylation. And as Varun reviewed quite at the beginning now, there's this whole explosion of biological clocks that started with a first generation chronological clocks, moving to the second generation aging clocks that are based on biological aging phenotypes. And again, the third generation, and in the second generation, I want to highlight, as we've talked to so far, the clocks of GRIM-age and pheno-age, which are the clocks based on pheno-age itself, so the DNA methylation predictor of pheno-age, and the GRIM-age clock, which is DNA methylation predictor of time until death, because we will be referring to those as we go along. But of course, along that path of all these epigenetic clocks, there's all sorts of clocks that have been created to understand systems of the body, et cetera. But I want to talk about what we now did with DNA methylation, just starting with that, and our EMR age variable. And that is, remember I said at the beginning, when we created EMR age, we were very strategic in that we looked at these 60,000 people in our creation of it, because they also had samples in the Mass General Brigham Biobank. So we took those EMR age and we identified 5,000 people in our biobank. And I pulled those samples and sent them to True Diagnostic, and we generated DNA methylation on these 5,000 people with the idea that we would then create the first EMR age DNA methylation predictor. And that's what we did. And I won't go into all the details of it, but this is, a very basic, AI predictor where we are doing, doing AI to create a DNA methylation predictor of our EMR age, and using lasso penalized regression of the CPGs. And I just want to show here that, with DNA methylation, the prediction and the accuracy of this is really quite striking, right? Our correlation in this predictive model is already over 0.9 to begin with. And so this is really exciting to see as the beginning of the study that we're already getting such good predictive accuracy with DNA methylation alone. And, DNA methylation is just one drop of blood. And that's an important biological metric. We've already talked about the fact that we know that, the epigenome is responsible for aging, and DNA methylation is one of the forms of epigenetics. But as Varun highlighted earlier, by golly, aging is a very complicated process. And while there's all these things we can look at here in terms of different ways that we know molecularly we age, we look at all of these things from telomeres to epigenetics to senescence, cellular senescence, etc., inflammation. We know that this is a very complex process. And while we're able to predict this very well with DNA methylation, is it important to consider other types of molecular data? And so this is really the next point of our query and quest, was to look at these individuals, these 5,000 individuals that we have DNA methylation on, and see if we added other molecular data, what we would then learn further. And what's the concept behind this, right? Well, the concept behind this is, of course, if we go back to, the theory of DNA going to RNA, going to proteins, going to small molecules, we know that all of these molecular data are interconnected and interconnected in important ways. And perhaps if we understand multiple levels of the molecular data, this can give us a much better perspective of what's happening to us as a biological system. And so let's just think about this. If we could consider the idea of, can we take genetics and can we add the epigenetics that we have? Can we add on gene expression through the transcriptome, where we see what genes are being expressed, to proteins, inflammatory proteins, all these proteins we know are good biological markers of aging, to small molecules through the metabolome. We know this is a major, some of the best clinical biomarkers, from blood glucose levels to measures of inflammation, LDL cholesterol, et cetera. And, while we didn't do all of this, I want to put it in this big framework, because as a researcher, this is how I'm thinking. Can we look at other things? Can we look at things like microbiome, gut microbial health, dysbiosis? Can we look at exposures that we've had and that are measured in our molecular profiles? And can we look at the phenome, what we're calling the phenome? And this also is reflected in electronic medical records, right? Can we look at the clinical data that we can extract from electronic medical records? And fundamentally, the idea of me as a researcher is, can we take all of this information and can we look at this all together in individuals at one point in time, synthesize that using systems biology, AI approaches, machine learning, to inform us about things like aging? And this concept, when we're looking at all of these data together, this molecular data, each one called its own omic. So together, the multi-omic can give us this multi-omic profile of individuals and help refine and understand why different people are aging at different rates. This is precision aging. This is what it's all about right here. And so while we can predict things, the concepts and the ability that we can do with all this multi-omic data is far, far beyond prediction if we can actually generate these data on everyone. So, okay, let's talk about the next thing that we did. Okay, so we have our 5,000 people with epigenetics, which is a huge number. We have, EMR age on all these people, plus a ton of clinical labs, clinical data, right? But what else did we, set out to do? Well, this has been sort of a mission of mine where I'm slowly trying to amass data within the Mass General Brigham Biobank system that has EMR data. And if you look at this over to the number of individuals within my own collaborative, my own lab, this is data that we have access to, which is clinical labs on, all of these people, 30,000 people over up to 30 years from the EMR data, whole exome sequencing on 60,000. And actually the clinical labs we actually have on 60,000 too. Transcriptomics, we don't have mRNA itself, but we have microRNA. We have our epigenetics, But for this project, what I want to focus on is the epigenetics, proteomics, and the metabolomics. And you'll see here over in the total number of individuals, our numbers for proteomics and metabolomics are higher than when we go all the way over to the left and we see the numbers of what we actually used for OMIC age. And that is because we are continuing to generate these other omics. So for what we're going to be showing you on OMIC age, we used the data that I have summarized over here to the left. 5,000 for epigenetics. Everyone has EMR age and a ton of other clinical data. We have 1,500 for proteomics and about 2,000 for metabolomics. So what did we then do? Now, we looked at how we created the DNAM model, predictive model for EMR age, and that's done, using sort of the standard approaches that have been used in this area of biological aging clocks. But now there's a few other quandaries, right? Because number one, we have a ton of data, but not only do we have a ton of data, the data all behave a little differently. And from a statistician like myself, it's not, we really can't just shove it all in a model. We have to be thinking about this in a systematic way. But I want to say here, if you look at this framework, the whole goal of what we want to do here is to create our new OMIC age predictive model using proteomics, methylation, metabolomics, and all the other clinical lab features that we have together with EMR age to create our overall OMIC age phenotype. Now, when we do this, I do want to highlight that this is a complex process. And when you start adding other omics, it becomes more and more complex. And this is an area that I think fundamentally it's important to us to understand that we need to go from something that's complex down to something that is simple. This, to me, is a crucial step because while we can use a lot of data to generate what we want to at the end of the day, what we generate needs to be something that we can measure very simply. And this created among us the concept of creating DNA methylation surrogates for our omic data. And by doing this, what we're able to do is take all of the omic data, combine it together in a way that we are then able to, at the end of the day, have one simple DNA methylation model that uses all the omic data. And let me show you how we do that. So I showed you we had all these omic data to begin with. And what we did was we said, okay, let's take all the proteomic data we have and all the metabolite data we have and a bunch of clinical data that we have in the EMR. And let's identify different types of metabolites, proteins, and clinical variables that we know are associated with EMR age. We can just do that through correlations. So once we get that list of proteins, metabolites, and clinical features, what we can then do through AI approaches, using things like lasso regression, we can create a DNA methylation surrogate of each of those things. So that's what we did. We took our DNA methylation data, we took our proteomic data, we combined it together, and we created DNA methylation surrogates of each of the important proteins, the important metabolites, and the important clinical variables. And as you see here, there are 107 proteins that were important. There were 267 metabolites and 21 clinical variables. And here's just examples of what I mean, just to put this in your framework so you can sort of understand this. Okay, so we have DNA methylation surrogates for things like, for proteins, we have things like albumin, IL-5, H1P, all of these different types of proteins. For metabolites, we have DNA surrogates of things like DHEAS, serotonin, cysteine, cortisol, uridine, carotene-diol, and DNA methylation surrogates for the clinical data and things like hematocrit, HbA1c, triglycerides, glucose. And so you see, if we did all of those surrogates, here's just an example of what those surrogates look like. These are the correlations of the surrogate with the actual value itself. We can now take all those DNA methylation surrogates, put them all in a model with a DNA methylation. And at the end of the day, when we do that, we can create our OMICm Age. Now, I want to highlight in here, I didn't highlight this to begin with, but in our model, we did want to strive for best in class, and we just published a paper on actually also incorporating better immune deconvolution with a 12-cell immune deconvolution. So we also refined even the way we are deconvoluting the immune cells in this OMICm Age. So now you have it. Now we have an OMICm Age clock. It's incorporating all the important proteins, metabolites, and clinical data with DNA methylation into our OMICm Age. And beautifully, we can now measure this on any individual just by running DNA methylation, which makes this test affordable. That is important, right? Because if we did all the tests, it is not affordable. But let's talk a little bit about how it actually predicts. And as you see here, our predictive accuracy is about it's 0.91 in the training, and then at the testing set here, it's also 0.91. So showing very, very robustly that it is predicting very well. So this is a good sign. But other things that are important is not only how well it predicts, but of course, what's more important is its relevance to disease outcomes. And here we see that when we look at this, the OMICm has the strongest associations with all of these disease outcomes. All-cause mortality, type 2 diabetes, stroke, CVD, depression, COPD, and cancer is the one it's not doing quite as well as. I think there are some reasons for that. But what I want to highlight here is if you look at this, the purple are our two clocks. They're DNA EMR Age and OMICm EMR Age. You see both of them here, and that in all these cases, it's actually regardless of which one we choose, we see that ours are performing far superior than any of the other clocks that are out there, whether it's Grim Age, Pheno Age, and a bunch of these other clocks by Hannum and Horvath. We see that we are having much stronger associations with clinically relevant outcomes. And that's important. But the other thing we want to highlight is really overall, if we're looking at things like mortality, which ones are best? And these are what are called ROC curves, and these are looking at both the sensitivity and the specificity for predicting mortality. And we have a 5-year mortality and a 10-year mortality. And basically, the further it goes up and over, the more accurate it is. And here you see, in our case, we have OMIC Age and DNA EMR Age here in purple and orange. And again, here we see it on DNA M in purple and orange. So by far outperforming all the other predictors. Now, so this really is encouraging that we see here now our OMICm Age is showing high predictive accuracy. Now, I want to highlight here that its predictive accuracy is pretty much on par with DNA methylation age. And that is actually important. And there's several reasons for that. I think one of them is that in and of itself, there's a lot of predictive value in DNA methylation. Another one is, I think there's other ways that we look at this where you see these differences are so small, where we see that OMIC Age is much better than DNA methylation, as in some of that disease outcomes. But I want to highlight even more why what we are doing with all these other omics is so fundamentally important, because we're just stepping at the beginning part of this. And I now want to go into other aspects of why this is, the OMIC Age is so important, and what else we're able to learn and take from this. And this is one of the things that Steve Horvath had said here, that this whole second section is on prediction. And we want to remember that when we are looking at a predictive clock, they are not always causal. What's in the predictive model is driven by AI, machine learning approaches, and they're not always causal. and extracting meaningful biology sometimes can be challenging. And, it is often that sometimes in these predictive clocks that the clinical implications are limited. And this is a major weakness of machine learning algorithms like what we're using in prediction. So that goes to the last part of what I want to talk about today, and that is understanding. And I want to talk now about how we can take all of what we've done to develop our OMIC, our DNA and our OMIC EMR age-based predictive algorithms in the context of what the importance of how we can now use omics to really, really ramp up our understanding, and why that is so fundamentally important in this next part of not only going from prediction, but going to the next step. And, there's a lot of companies now that do predictive algorithms, right? And I don't think any do them as best in class as what I've shown here today, but prediction is one thing, but taking it on to this next part where we really can extract important biological meaning is critical. And this is where we are now doing this in a way that I just haven't, this has not been done, and we're really on the realm of new discovery here. And when we think about this, right, we've now created, this EMR age predictive models. We take a drop of blood, we measure, your omic age, and we can immediately give you a good sense of what your overall biological age is. But can we identify causes of biological aging? Can we prevent biological aging? Can we reverse it? And if we just stopped at prediction, the answer is no. Unfortunately, we can't. We may, in terms of general things, you can exercise more, you can eat better, you can do this, but it's all general. This talk is about precision aging. It is not about aging in general. So what we now want to talk about is how we can now take this and understand this on a precision aging framework for each individual, right? And that is what we want to do. Can we actually look at this and give you the specific advice, or your patients the specific advice of what they need to do? And so that goes into this idea of really characterizing real effects that we see with DNA methylation. And I talk about this idea that, when we take DNA methylation or a lot of the other types of epigenetics, it is very often hard from going from that to understanding. And the key reason is because we're just looking at hypermethylation, that's not giving us a lot of information. But don't forget, in what we've now generated, we've generated metabolomic data, proteomic data, and we have clinical data. We have all sorts of rich information now that we have DNA methylation surrogates for. So for anyone who comes in, right, and you get your omic age done, we also have methylation surrogates that give us a lot more information about what's actually happening biologically. And with that, we can, the important thing is with things like proteomics and metabolomics and clinical data, that is really what we need to bridge the gap between methylation and aging. Now, putting this in the broader framework of how do we do that? So let's think about how we can use those omics to bridge this gap in our understanding from what we see with epigenetics to what we understand about aging, with things like the proteome and the metabolome. Well, we can take those, and now with what we've done with omics, we have a very good sense of what the proteins and metabolites are doing and their individual associations with EMR age. We can see that now. We see things that have strong associations with EMR age and whether they're increasing it or decreasing it. And, we can see things like metabolites in the energy cycle, metabolites of frailty. We have specific biomarkers like this one, vinonylic mandelic acid. We see things with dysbiosis of the gut microbiome. We see different markers of kidney disease. We see different inflammatory cytokines. And the point is we can use all that now and understand how that relates to DNA methylation. I also want to highlight here, not only can we understand those molecules, but we can also understand a lot of deleterious exposures and things like PFAS or other exposures that we know are bad for aging. And And now with what we now have with our omic data, we can actually say, we can look at these different individual omic variants, whether they're proteins or metabolites, and we can say, and as you see here, if you look at the picture as I do this cartoon going forward, you can see how I'm trying to put together understanding with the molecular data with DNA methylation. PFAS may increase inflammatory signals and increase aging. Good microbial metabolites decrease aging. Cotinine is associated with increased aging. It's a biomarker of smoking. Endogenous steroids reduce levels, increase levels, decrease aging. Good food, things like L-serine and omega-3 fatty acids are going to decrease aging. Other biomarkers we know for aging are going to be associated with increased aging. Serotonin is going to decrease aging. DMTPA is going to increase aging. And these are just a very select group of the overall molecules that we saw that have strong associations with EMR age. And from this now, we have a much better global picture of how we can understand what's happening with your epigenome and the different ways that biologically we are seeing associations that then we may be able to do further research and understand causation. So put simply, omics bridge the gap from DNA methylation to biologic understanding. And when I talk about this, I want to talk about this, that this is all now something that, well, we did this and we mapped this all together with the omic data we have in our cohort. And we will continue mapping that. Trust me, we aren't done. When we go to each individual, we can measure surrogates of these things in each individual. And that gives us an idea for each individual of how they uniquely may have contributors to how they are aging. And this is a very simple way to understand and extend our biologic understanding. So we've now gone through and we've seen how we can create a biological aging phenotype that is best in class by putting together both the clinical and the mortality data together, wellness and mortality, to create our EMR age. We've shown how we can predict it with not only DNA methylation, but also putting in these DNA methylation surrogates of the other omics to create this multi-omic aging predictor. And we now see how we can use all of those omics to really further our understanding of the disease processes that are impacting our epigenome. Now, I want to highlight that we are like just at the beginning. There's so much we can do with the data that we haven't even touched upon, that our understanding is really at the beginning. And I think this field as a whole, there's a lot of understanding that's really at the beginning. And I'm excited to see what we continue to learn as we continue to delve into the multi-omic data that we have. But I want to go back to this, right? Because this all comes back to the idea of precision aging. And we've looked at all this data, and what I want to highlight now is, when we are looking at what we have now with omic age and the omic data and DNA methylation surrogates that we have developed, we are really uniquely positioned to take each person and ask from a precision aging framework what their EMR age is and what is being affected for them that may be driving this, not only from prediction, but also going more into association and hopefully leading you even further into causation. And it's really about then getting to this EMR picture I see here, where we can then look how we can look at the totality of health in aging and optimize it with our aging predictor and omic data. So with that, I'm going to stop. I know I went a long time. Apologize for that. But I'm going to head it over to you, Ryan, and Varun, for either questions, and just highlight here, there's a massive amount of people working on this, and just highlight the amazing work by Varun and Qingwen Chen, who led a lot of the data analyses in what I showed here.
Yeah, thank you so much, Dr. Sue. And I'm excited to jump in here a little bit just to talk about, I think, something different than the philosophy of how we're approaching this problem, but really to try and approach the clinical picture. True diagnostics is really unique because 90% of what we do is still to integrative medicine physicians. And so we really get to see on a patient-specific level how this is impacting individuals. And I think hopefully what we'll talk about today is why this is going to be a better experience for any clinicians who have used our test before, and exactly how it overcomes some previous barriers. So we won't spend a ton of time on this. I'll try. I know that everyone is probably anxious to spend the rest of their night, but I think this is where the rubber meets the road. And so there are always these issues with previous epigenetic clocks, which we really want to improve. One of the big issues that has been thrown around for a long time is the precision of these clocks. Oftentimes people ask us, how often should we test? And a lot of times with even some of the earlier algorithms, this was a huge limitation for us, because if we were to take even the same sample with some of these earlier age clocks, they could differ for up to several years. And I apologize, my screen share is not loading at the moment, so I'm going to try one more time. We'll see if this is working. So unfortunately, my screen share is having a few issues. We'll still work on it, but in the meantime, I don't want to keep people waiting. And so as we're talking right now, I just want to talk about this precision. Right we want to get the exact same score. And with previous clocks, if we were doing this, we could get up to 50% differences in these epigenetic clocks, which makes it virtually impossible to do on an individual level within a period of a couple of years. Some of these early clocks could vary up to by 3.9 years for each sample, meaning that if you're testing in a period of 3.9 years, you don't know if it's true accuracy or if it's just variation within the measurement. I'm happy to say that with OMIC Age, one of the other issues and problems that we've solved is that our precision is excellent. If we're measuring the exact same sample, we have less than really a 0.5% variance in that exact same sample. And so this is very precise, meaning it can be measured. And if we see change, we're fairly confident that that is true biological change as we're measuring the methylation and not just noise in the measurement. So definitely something that we're happy to have with this clock. One of the other big problems that has been happening with these clocks has been the variation that can happen with immune cell subsets. With epigenetics, every single cell has a different epigenetic expression, which also gives it its cellular identity. And so one thing we don't want to happen is for someone to have immune cell subset changes, such as, let's just say they start drinking caffeine, their naive CD4 cells go up, and then that drastically changes their biological age. We know that that wouldn't be true, right? If simple behavior is changing their biological age significantly, that's a pretty big problem. And what we've seen is that, for instance, even in naive human CD8 T cells, they can be up to 20 years different with the same algorithm of cells of a very similar type. And so we really needed to solve this problem. Dr. Su and Varun have both already put some references about how we did this with our immune deconvolution method. But now we can actually predict immune cell subsets with essentially the same resolution of things like RNA-seq data or flow cytometry data. And so now we can actually include this into our algorithm and make sure it doesn't vary as these immune cell subsets change. And so already we've eliminated two of the major problems with some of these epigenetic clocks. One of the other things that I think really helps clinically is demonstrating the importance of risk. And this is something we've also made an effort to do, is to translate how someone's aging might increase or decrease their risk. This is a slide we always like to show, a variation of a slide that Matt Caverline has posted often on Twitter. But this is a slide which shows the top 3 causes of death in 2020 and shows the relative risk factors that contribute most to that cause of death. And as you can see, aging dwarfs things that we already know and most everyone would know are really bad, like smoking and obesity. And so we really wanted to have a way to show this to patients as they were taking our tests, how their aging rate is increasing or decreasing their risk using the assumptions that we had in this cohort from Brigham's. And so on this report now, we're actually going to show how much a 1-year increase or decrease would increase or decrease relative risk as a result of epigenetic change. And we're hoping that this will also help patients to get really excited about driving outcomes lower. And so we've solved the immune issue, we've solved the precision issue, and hopefully now we can help give more evidence on why this is important clinically in terms of eliciting change. One of the other big issues that a lot of people have had is that sometimes these age biomarkers don't match up with the other clinical biomarkers that you're testing. So for instance, you'll have someone in your clinic that is overweight, has insulin resistance related issues, has been a smoker their entire life, likes to drink, doesn't do physical activity. And they might come out with some of these earlier age clocks as being very young. And that doesn't match up with this clinical picture. And in fact, might even reinforce some of those negative behaviors that you're trying to stop in the clinic. The one clock that is great for this and always shows consistency with the larger clinical picture has been that Dunedin pace. But oftentimes those first generation clocks still aren't compatible. And what we're seeing now in this correlation chart, if you look down at the bottom, is that we're starting to see that our OMIC age has incredibly good consistency with that Dunedin pace. But in addition to that, what we're also seeing is that just from a sort of a back of the hand measurement check, this is much more in line with the clinical phenotypes that you might be seeing within a clinic. And so we think this is very, very helpful because it is consistent information that backs up all of the classical clinical thinking and knowledge that you're applying to your patients. And we think this is very, very important. But in addition to that, as Dr. Sue already mentioned, we're also seeing this proven in outcome data as well. So not only is it just better by looking at the patient, seeing more compatibility, but in addition to that, we're also seeing that, again, it outpredicts any other epigenetic clock in a host of outcomes. Really, GRIM-age and COPD are the only exception to this, where GRIM-age is still great at COPD, especially we believe it's because of that great smoking pack year prediction that it has within that as a methylation risk score. But really outside of that, OMIC age is really outperforming all of these clocks and capturing a multitude of risk. To sort of also talk to Dr. Kara Fitzgerald's question about the difference between EMR age as a methylation predictor and OMIC age, it's the OMIC age included those methylation risk scores. So those surrogates of methylation like HbA1c, C-reactive protein, blood urea nitrogen, all of those are included as features in the OMIC age algorithm. And so we're capturing multiple pieces of biology of aging. And when we're doing that, we're better at predicting not just time until death, but we're better at predicting all of these other different disease categories and really representing this whole bigger picture of biological aging. And so what I want to talk about next is also the piece I think is very exciting. Usually if someone had a fast pace of aging or was accelerated on these biological clocks, we couldn't really tell you why. And that's been one limitation that is very frustrating clinically. You might have a patient who's aging and not know the reason why we're seeing those different features. Now we're actually able to add a lot of resolution there. So as you'll see on this graph on the right, this graphic on the right, we have all of our methylation risk score surrogates, which are actually included into this algorithm. And there are 36 of these, which are included as outputs on our reporting, where we're actually going to be quantifying the relative levels of hematocrit or hemoglobin or uridine, for instance. And in addition to that, we're also going to be showing you the relative impact to OMIC age overall. So if you look at that bottom sort of image there, you can see the clinical factors in methylation risk scores, which are input into this algorithm. So we're actually going to be able to tell you that, for instance, here, albumin is the most protective feature of this person's individual methylation risk scores. We can also tell you that the mean red cell distribution width and HbA1c are going to be the biggest negative impacts. And so what we can start to do is classify these different methylation risk scores and get a better clinical picture of what systems are causing this aging process. So for instance, out of all of the classifications we did, we saw really three big areas which were highly represented. That is cardiovascular factors, which really represent a lot of lipids. We see endocrine with type 2 diabetes and insulin resistance, and then we see a lot of inflammatory markers. So these tend to be the cardiovascular system with inclusion of lipids, the inflammation markers, and then endocrine markers tend to be the highest and most well-represented methylation risk scores which were included. And these were selected for their ability to, again, predict that time until death marker. And so we're going to separate this on the reporting into three different categories. It'll be all of the clinical markers, all of the proteomic, and all of the metabolomic. And what we're really hoping to do here is provide a lot more resolution into the why. Why are we seeing increased aging? And now that we also know that and have proven that it's the most predictive of all of these different outcomes of all the clocks, we hope we can change these factors and then see changes in the overall aging rates as a way to really influence these aging rates in a preventative fashion. And so it'll look like this on the report. This is actually just a direct screenshot of an individual value where we can see blood urea nitrogen, HbA1c, alkaline phosphatase, and fasting glucose glucose, and we're going to be able to estimate those relative proportions. It's also important to mention that these are not going to be as accurate as the clinical labs that you're getting directly. This is a DNA methylation prediction of these values, and all of these are fairly accurate. They're still not a direct replacement for the clinical markers you're already getting. We hope that one day they will be, but right now it's just meant to quantify the relative impact of these different systems onto your overall biological aging. In addition to this, we've also done some analyses which can connect these methylation risk scores outside of this omic age, but independently to different diseases. And so in this image, what you can see here on the outsides are methylation risk score surrogates. So everything from blood urea nitrogen, glucose, lung function measurements, and you can see they're connected to these different diseases. In the red lines, we see generally that if a value is increasing, it's also increasing risk of that disease in the middle. If we see green, it's the opposite or inverse association. And the thickness of the line is going to be the effect size to that particular disease status. And so, for instance, for glucose, you can see its biggest line and connection is to type 2 diabetes. Again, this is outside of omic age just generally and just using a methylation risk score for glucose. And so what we're seeing is really matching up with what we know clinically, which is that many of these metabolites are connected to these processes, and we're seeing high degrees of association. So, for instance, if we look at FEV1, we see it's connected to COPD. If we look at glucose, we see it's connected to type 2 diabetes, and the effect sizes make sense, which again is showing consistency among the classical clinical picture that we might see from these EMRs, but using DNA methylation as an independent surrogate. However, we can do a lot more with this. We recently did a trial with Stanford where we looked at 21 pairs of twins. One twin was put on a vegan diet, the other was put on an omnivore diet. And so we used all these methylation risk scores that we created, which are over 400, and that's out of the 30,000 possible methylation risk scores we are going to create from all the metabolomic and proteomic data that we have. But in this analysis, we wanted to look at what methylation risk scores were changing in this clinical data set. And what we found was that there were 3 significant methylation risk score surrogates which changed. These are here, and you can see their common names. So things like ribonuclease from the pancreas or CXCL4 in terms of inflammatory markers. And what we saw is that these changes that we detected in these groups between this vegan and omnivore diet were well described in previous literature before, which again backs up the fact that we're seeing interventional change that is consistent with interventional change measured by those direct metabolites. But instead of having to measure each of those direct metabolites, what we're able to do is actually just use DNA methylation as our sole marker to predict many of these other different outcomes. And together, we're hoping that as we continue to add methylation risk scores, we'll get much more information about the clinical changes and really get to that level of personalized medicine. And so again, I just wanted to talk about this a little bit more. So here in particularly this patient, these are the methylation risk scores that we're going to be showing clinically. We've already talked about things like albumin or HbA1c. And what this backs up is, again, previous assumptions that we've seen before. So we actually can see, for instance, the time until death with looking at albumin levels. And we see that albumin is weighted heavily in this model, which again backs up these assumptions. And again, it's very close to the clinical picture you'll be seeing. We also can do this with metabolites. So for instance, one of the best weighted metabolites is uridine. Secondarily to that, one of the protective metabolites is keratine-diol. Both of these can actually be done via supplementation. And we've actually seen in very small, in a really 1 or 2 cohorts, that supplementation with these cofactors like lutein or uridine actually change these methylation risk scores directly and therefore also impact omic age. We need a lot of validation to do this a little bit further, but it's also the exciting part as well, because the algorithm for predicting time until death actually selected the relevant metabolites. And then it also selected their clinical weights, as you're seeing here. So we can actually, again, see which is the most important to our aging and uncover unique associations. So in the case of uridine, this is never a molecule that I've seen anyone test clinically. But if we look at the body of literature around uridine, what we see is it is highly connected to aging. Here is just one journal article where we can see that intestinal aging is led by uridine. We see in several other studies that uridine is a classical aging-related biomarker that can help improve stem cell mobilization, stemness of stem cells, and help with regenerative repair and recovery. In several other studies, it's been associated with risk of all-cause mortality. And we can't really tell you the exact mechanism, but we can tell you it's obviously important. And now we can actually even quantify those relative levels within a patient. So again, we're starting to uncover unique and novel associations, but we're also able to report it back on that report. Carotene or lutein is another good example. Most people are probably familiar with this as a carotenoid, which is in spinach, kale, and yellow carrots, that has been classically associated with eye health. But recent studies have been showing that it also helps reduce metabolic syndrome, coronary heart disease, and is highly related to inflammation. And this is not the first time these epigenetic clocks have shown the importance of carotenoids. So for instance, even in this image, you can see the impact of carotenoid levels in the blood and its association with better DNA methylation GRIM age. And so again, carotenoids have been selected and once again show the importance where we can quantify its relative levels. And so these are just some metabolomic examples, but I also want to give an example of one of these proteomic values. And we'll do a lot of education on this in future topics of conversation, where we'll actually go into each of these 36 metabolites or proteins or clinical values and give education about what we know about them as biomarkers, and also what changes them as biomarkers, so that we might be able to sort of speculate on different types of interventions which might change these things. And so again, here we see positive associations for things like carboxypeptidase B2 and the peroxidase as well, which we'll talk about a little bit more. And we see negative associations between like heat shock protein 70 and an IGF-1 binding protein 2. In the case of the serum peroxidase and the aryl esterase 1, we know that this is actually an enzyme which is responsible for the major anti-atherosclerotic component of HDL cholesterol. So again, going back to that cardiovascular relationship to lipid levels, this is a very important factor which we actually want to increase. When as we get older and this becomes impaired, we see a much higher risk of cardiovascular and cardiometabolic diseases. And so everyone obviously knows that HDL is protective, and this is one of the mechanisms that it actually exerts that effect, and this is why we're seeing it to be weighted so highly in our OMIC Age algorithm. And so overall, we're going to now start to give a resolution which previously we just didn't have with these epigenetic clocks, where we couldn't answer the why while still improving on all of the features which have been problematic in these clocks in the first place. And so we're really excited. I think these methylation risk scores also unlock the potential of DNA methylation as biomarkers outside of aging. Obviously, aging has been our major focus, but these methylation risk scores allow us to do more. And I want to cite another good example of this. This is from a UCLA-related trial where they showed that incorporating methylation risk scores into these EMRs significantly improved outcomes in 139 different outcomes, while regular polygenic risk scores only improved 22. And the magnitude of change was significantly higher for these methylation risk scores. And I think that this shows the benefit and the clinical utility of these methylation risk scores, where we're not just looking at the DNA sequence for what can happen, we're looking at the regulation of gene components to give us a better idea of what's happening now and what we can do about it for the patient. And that gives a lot of clinical utility we just didn't have with some of those polygenic risk scores. And to end, I just want to show a good example of how this has impacted methylation clocks previously. If we've shown this GRIM age image several times, where they actually use DNA methylation risk score surrogates for plasma proteins. In GRIM age 2, they actually added 2 methylation risk score predictors. They added HbA1c and they added CRP. And by doing that, GRIM age 2 is significantly better at predicting almost every outcome than regular GRIM age. And I think that this is exactly what we're doing. By adding these methylation risk scores, which are able to pick up biological context outside of just aging, we're able to capture the full risk factors of human health and consolidate it into one number, which we can hopefully effectively change through clinical intervention. And so I think at the end of the day, what we're saying is that we've worked really hard for this OMIC Age algorithm to compile the biggest biological signal possible. And together, now that we have this, the outputs of this also give us a lot more information about what is causing this aging process than previous methods. And together, we hope that these entire suite of methylation diagnostics will continue to improve healthcare and personalized medicine. So with that, I would like to open it up to any type of questions that might still be remaining. I know that people have been asking questions as we've been going along, so there might not be many, but excited to dive into